Dear students, myself, Dr. Zubaria Hamjaved, a lecturer in Botany Department of Microbiology, BOTN 4128. Today, here is your lecture number 24, and we will discuss about conditions influencing the effectiveness of antimicrobial agents. First of all, see the contents of today's lecture. In the beginning, we discuss some terminologies and then the mode of action of antimicrobial agents, conditions influencing the effectiveness of antimicrobial agents, and which we will discuss about some population size, population composition, concentration or intensity of an antimicrobial agents, and then duration of exposure, and at the last we discuss about the effect of temperature on the effectiveness of antimicrobial agents. First of all, we will discuss some terminologies which is necessary to understand before starting our today's lecture. As you all know about sterilization, disinfection, sanitization, and some other terminologies, but today we will revise our terminologies. As we all know that terminology is especially important when the control of microorganisms is discussed, because words like disinfectant and antiseptic often are used loosely. The situation is even more confusing because a particular treatment can either inhibit growth or kill the microorganisms depending on the conditions. First of all, sterilization, which is derived from the Latin word sterilis. It means unable to produce offsprings. Process where all living cells, viable spores, viruses, and viroids are either destroyed or removed from an object or habitat or a place, totally free of viable moss, spores, and other infectious agents, or all type of microorganisms. Sterilants. Sterilization by a chemical agent is known as sterilant. Then disinfection. Disinfection means the killing, inhibition, or removal of most may cause disease. Means most of the substances may remove or inhibit or kill that may cause disease. Here we have a goal to destroy the potential pathogens or reduces the total microbial population. Disinfectants or agents, usually these are chemicals, carry out disinfection used on inanimate objects. As for example, a disinfectant does not necessarily sterilize an object with viable spores, and few moss may remain. It is not effective for disinfection. Disinfection is not effective for all type of microorganisms, but it may be used, or it is commonly used for those substances which through which we work, like our table, our floors, our windows, etc. Then sanitization. The microbial population is reduced to safety levels for the public health standards. The inanimate object is cleaned and partially disinfected, as for example eating utensils in our restaurants or in our places outside the working places. Then some sanitizers. We also use sanitizers to sanitize our working substances or our hands. Then antisepsis prevention of infection or sepsis and accomplished with antiseptics. Antiseptics, these are chemical agents applied to tissue to prevent infection by killing or inhibiting pathogen growth. They also reduce the total microbial population, not as much toxic as disinfectants because cannot destroy too much host tissue, but it is effective in controlling the microbial organisms or in controlling the microbial growth. Therefore, we also use antiseptics and antisepsis in our aseptic techniques. Then fixes. So fixes means denotation of antimicrobial agents. Like we use side, the word germicide, the antiseptics, statistic, or antimicrobial agent. Where we use side, it means substances that kill the microorganisms, and it is also derived from the Latin word which is sida, which means kill, to kill. Then germicide. Germicide means kill pathogens and also non-pathogens, but not necessarily endospores. And then our antiseptics or disinfectants. These can be effective against a specific growth like bactericide, fungicide, algicide, or viricide. When we use the word static, it means do not kill but prevent growth. If removed, growth will be resumed. Bacteria, as for example, like bacteriostatic or fungistatics. Then here is some resistant or most resistant and least resistant microorganisms examples, in which we see that the endospores of bacteria are the top of the resistant microorganism category, and the viruses with lipid envelopes are at the least resistant category. And here we also, from top to bottom, we have endospores of bacteria, then mycobacteria, cysts of protozoa, then vegetative protozoa, gram-negative bacteria, fungi, including most fungal spore forms, viruses without envelopes, gram-positive bacteria, and at the least resistant microorganism or microbial category, we have viruses with lipid envelopes. When we see antimicrobials, antimicrobial agents, an agent that kills moss or microorganisms or inhibit their growth. Here some categories of antimicrobial agent. We have antibiotics or antibacterials, which works against the bacteria, as for example drugs for bacterial pneumonia, and also antivirals that work against the viruses, as for example drugs for herpes virus and HIV, human immunodeficiency virus. Then antiparasitic agents, which works against the parasites, as for example drugs for malaria. Then antifungals that works against the fungi, for example drugs for yeast infections. Here we have the mode of action of antimicrobial agent. The antimicrobial agents work with three different mechanisms or three different principles, that either they affect the plasma membrane, or they affect the proteins, or they affect the nucleic acids. In this figure, that they may affect the cell wall synthesis, they may affect the nucleic acid synthesis, and also the protein synthesis. And here are some examples of my antimicrobial agents. The agents which affect the cell wall synthesis include beta-lactams, vancomycin, bacitracin, And the agents that affect the nucleic acid synthesis include the DNA gyrase, DNA polymerase, etc. And also some examples of protein synthesis effectants. When we see the antimicrobial agents which affect the plasma membrane, they actually damaged or cell content leaks into the surrounding medium. And when we see the proteins, then we see that they affect the enzyme active sites and cause them inactivated. And sometimes they also cause cell denaturation. They may also change the shape of the cells into different shapes and also blocking the active sites. And when we see the nucleic acids, then radiation or some chemicals lethally damage the DNA or RNA. Microbes can no longer replicate in this situation. And sometimes we also use the UV radiation, which causes formation of dimers between two thymine bases. And this figure is also describing the same mode of action of antimicrobial agents. In the first figure, you see that normal microbiota keeps opportunistic pathogens in check. And in the second category, you see the broad spectrum antibiotics which kill non-resistant cells only. And at the last, you see the drug resistant pathogens proliferate and can cause a super infection. Now, here we will discuss some conditions influencing the effectiveness of antimicrobial agents, in which we will study, first of all, we will study about the population size, then population composition, and then concentration or intensity of an antimicrobial agent. And on the fourth number, we will discuss duration of exposure, and then temperature, and at the last some local environmental conditions which affect the conditions which affect the effectiveness of antimicrobials. Now, first of all, population size. We should remind that what is population size? Population size is defined as the number of individuals present in a subjectively designated geographic range. The population size, because an equal fraction of a microbial population is killed during each interval, a large population requires a longer time to die than a smaller one. This can be seen in the theoretical heat killing experiment shown in previous table and figure, as we see in the table and figure of our last lecture, in lecture number 23. The same principle applies to the chemical antimicrobial agents. Second one is population composition. Population composition, the effectiveness of an agent varies greatly with the nature of the organisms being treated, because microorganisms differ markedly in susceptibility. As we all know that bacterial endospores are much more resistant to most antimicrobial agents than are vegetative forms, and the younger cells are usually more readily destroyed than mature organisms. Some species are able to withstand adverse conditions better than others, but not all the microorganisms withstand the adverse conditions. For instance, as we see that Mycobacterium tuberculosis, which causes tuberculosis, is much more resistant to antimicrobial agents than most of the other bacteria. About the concentration or intensity of an antimicrobial agent, we should discuss about some chemical agents or physical agents, because the concentration or intensity of these agents may cause a major effect on the antimicrobial agents. Often, but not always, the more concentrated a chemical agent or intense a physical agent, the more rapidly microorganisms are destroyed. However, agent effectiveness usually is not directly related to the concentration or intensity. Over a short range, a small increase in concentration leads to an exponential rise in effectiveness. Beyond a certain point, increases may not raise the killing rate much at all. Sometimes an agent is more effective at lower concentration, but sometimes an agent is not more effective at lower concentration. As for example, when we see the 70% ethanol, then 70% ethanol is more effective than 95% ethanol because its active is enhanced by the presence of water. So we can't uh, mm, we can't say that concentration or that more the concentration of our agent will create more effects. Then duration of exposure. The longer a population is exposed to a microbial agent, the more organisms are killed. To act to achieve sterilization, an exposure duration sufficient to reduce the probability of survival to 10 ratio to 10 raised to power minus 6 or less should be used. As we see from the figure, that when we plot a graph between time in minutes and percentage of death rate, then we see that with the passage of time or by increasing the time, the death rate is also increased, where the A shows the lag death phase and the B shows the log death phase and the C shows the stationary death phase. Fifth one is temperature. An increase in temperature at which a chemical acts often enhances its activity. Frequently, a lower concentration of disinfectant or sterilizing agent can be used at a higher temperature. Here you see in the figure that there are some bacteria which have lower growth at lower temperature and higher destroy ratio at higher temperature. But there are also some bacteria which show moderate or rapid growth in this zone of temperature. Then there are some local environmental factors also which affect the conditions or which affects the effectiveness of our antimicrobial agents, which includes organic matter and other factors. The population to be controlled is not isolated but surrounded by environmental factors that may either offer protection or aid in its destruction. As for example, because heat kills more readily at an acidic pH, acidic foods and beverages such as fruits, tomatoes are easier to pasteurize than foods with higher pH. As for example, like our milk. A second important environmental factor is organic matter. This organic matter which can protect microorganisms against heating and chemical disinfectants. And when we see about the biofilms, then biofilms are a good example. The organic matter in a biofilm protects the biofilm's microorganisms, and the biofilm and its microbes often are hard to remove. Furthermore, it has been clearly documented that bacteria in biofilms are altered physiologically, and this makes them less susceptible to many environmental or antimicrobial agents. Because of the impact of organic matter, it may be necessary to clean objects, especially over syringes and medical or dental equipments that should be disinfected or sterilized before they are used. The same care must be taken when pathogens are destroyed during the preparation of our drinking water. As we see in our city's water supply, that when a city's water supply has a high content of organic matter, steps are taken to decrease the organic matter or to add some more chlorine. Discussing all these six conditions that influences the effectiveness of the antimicrobial agents. We will study the methods of control in our next lecture, in which we will study some mechanical and chemical factors to control the microbial growth. And then we will see we will study about some physical agents, some chemical agents, and then mechanical removal methods. So class, these are all the references from which today's lecture is prepared. If you want to study some more material, then you may also read the chapters available in these books, as for example from this Surinder Kumar textbook of microbiology, from the essential microbiology, and also from the principle and exploration of microbiology. So dear class, if you have any question relevant to our today's lecture, then you may freely ask me. Thank you.